everyone, and welcome to our lesson on the light-independent reaction, which is the Calvin cycle. Now, I really like this picture here to depict the Calvin cycle because although sometimes some sources may call this the dark reaction because it does not directly require sunlight for the Calvin cycle to operate, it still does require the products from the light reactions to actually facilitate the cycle to occur. So this is why I like that one light bulb on to remind us that it still does kind of depend on the light, just indirectly because it depends on the light reactions occurring to generate specifically ATP and NADPH to get that cycle going. So let's take a look. Here is a big overview of the Calvin cycle. You can see that there are three main phases. There is the carbon fixation phase, and you can see on the top, kind of at 1 o'clock there, carbon dioxide is coming in. Okay, so carbon dioxide is definitely a requirement for this process to occur. There's a reduction stage, and you can see ATP is coming in and leaving as ADP. There's NADPH coming in and leaving as NADP. So there's a series of reactions that involve redox reactions with these high energy molecules. And then stage 3 is the regeneration. To be able to come back and start the cycle again, we need to regenerate the molecule that comes back up as rubisco at the end, because that is the molecule required to start the cycle once again and to interact with carbon dioxide. Through that process, you can also see some ATP coming in and leaving as ADP. Okay, so we see some redox happening there as well. So overall, we've been looking at the different stages of photosynthesis. Remember stage 1, that was photoexcitation. Stage 2 was the electron transport chain and chemiosmosis that we saw occurring within photosystems 2 and 1 within the thylakoid membrane there. And stage 3, this is the Calvin cycle, and this is occurring in the stroma of that fluid like substance, the stroma that is within the chloroplast. So let's put ourselves in the mindset of a plant. So we need to produce all of our organic molecules necessary for growth. We need to produce our carbohydrates, our lipids, our proteins, our nucleic acids. We're not heterotrophic organisms. We can't just go to the fridge, open it up, and have an array of nutrients in front of us. We have to do it all ourselves. So we need to store our chemical energy, that ATP, and we know that ATP is produced from light reactions to be used towards the synthesis of these organic compounds like glucose. And this ATP needs to be stored in a stable form. It must be able to be moved around the plant and be saved for a rainy day. What happens when the sun is not out? What happens when it's overcast? Okay, we need to have that energy storage available to enter the Calvin cycle should the light reactions not be simultaneously happening at the same time. So again, there's another overview of the equation of photosynthesis. So carbon dioxide, we're going to be seeing coming into play today in the Calvin cycle. Water, we saw the necessities of water yesterday in the light reactions of that photolysis, right? We get light energy. Sometimes it's shown over the arrow, but it's shown here in the reactants, which makes sense because it's absolutely necessary for photosynthesis to occur. And in the end, we're going to have glucose and oxygen. So we have the oxygen already from the light reaction. We're going to be talking about the building of glucose today. So remember, the light reactions convert solar energy to chemical energy, right? We can't just use solar energy directly in the Calvin cycle. We first have to convert it to its usable form in the cell, which is ATP. Let's not also forget about NADPH. This is that high energy molecule that is a precursor to ATP often, but also can be used to reduce other molecules and transform them in the process. So we'll be seeing that today. So remember, NADPH is money in the bank when we're talking about photosynthesis. So from the light reaction, what can we do now? We are going to build stuff today. Photosynthesis. Synthesis means building that anabolic reaction, and we're going to be building glucose molecules. So we want to make C6H12O6 through the process of synthesis. So how are we going to do this? From what? What raw materials are available? Okay, and we're going to be looking at the first stage in a bit, where we're going to be seeing CO2 coming into play. It's going to enter through the stoma of the leaves. Okay, this is through the process of carbon fixation, which we'll talk about. We're going to be seeing NADPH coming in and leaving as NADP, which reduces CO2 in the process. Look at all the details, okay? And in the end, we'll also be seeing ATP coming in as well. But in the end, we're going to be forming this large glucose molecule, and that carbon is coming from the carbon in the carbon dioxide. So how does it get there? We need 6 of them per glucose molecule. So CO2 has very little chemical energy. It's fully oxidized. It's a smaller compound versus C6H12O6. That's a very large molecule. It's highly reduced. Okay, it has a lot of chemical energy. Now, when we're talking about synthesis, such as photosynthesis, it's an endergonic process. We're building things up. This takes a lot of energy requirements to do so. Okay, so we're going to be taking in energy. And we saw that already in the light reaction as it took in those photons from the sunlight. So in the Calvin cycle specifically, we're going to be looking at the reduction of CO2 and how it becomes C6H12O6. And there's many small uphill steps. And when we're talking about uphill, that means that the potential energy is growing in that growing molecule as bonds are formed and C6H12O6 eventually forms from that smaller energy molecule, which is CO2. So each small step is catalyzed by specific enzymes, and it uses energy that is stored in ATP and NADPH, which came from the sun originally, right? From the light reactions. So going from the light reactions to the Calvin cycle, we are now moving out of the thylakoid, and we are going to be seeing a Calvin cycle occurring in the stroma of the chloroplast, that fluid-filled area, similar to the cytoplasm of the cell, but we're still in the chloroplast here. And we need products from the light reaction to drive the synthesis reaction that occurs during the Calvin cycle. So we made that ATP that is generated from the light reactions, as well as the NADPH. Okay, so we're going to be seeing the start of the Calvin cycle, and then we're going to be seeing like a midway point where it actually produces a very important molecule that together, through a combination of this cycle happening multiple times, it forms glucose in the end. Okay, so rubisco is an enzyme required to start this process, and it catalyzes RUBP to bond with the carbon dioxide and form that first new molecule in the Calvin cycle. And then through a series of redox reactions, we're going to end up getting G3P, which is that molecule circled down at 6 o'clock there. And this G3P is very important. This is the product of the Calvin cycle. okay? And by little bits and little bits of G3P created as the cycle turns and turns and turns, we end up getting the building blocks for glucose, which in the end bonds together and forms that large glucose molecule. So each turn of the Calvin cycle fixes one carbon. For the net synthesis of one G3P molecule, the cycle must take place 3 times, fixing 3 molecules of CO2. To make one glucose molecule would require 6 cycles and the fixation of 6 CO2 molecules. Let's take a look on the next page of all this beautiful detail.
Okay, so this is the tell-all of the Calvin cycle, of the light-independent reaction. We don't see light energy directly hitting any areas here, okay, although we are using the ATP and the NADPH from the light reactions. So where do we start here? I've divided this entire cycle into the 3 phases. Okay, so let's start with the carbon fixation phase, which I've put in purple, and those are numbered as 1 and 2. Okay, so carbon fixation, to summarize it, it captures CO2 from the atmosphere and it makes it into a usable molecule in the Calvin cycle. So 1, you can see that the Rubisco enzyme combines CO2 that's coming in at the very top there with a molecule called RUBP. And this forms a highly unstable 6-carbon molecule, so unstable that it barely retains itself before it splits. So number 2 is when it splits, that unstable 6-carbon intermediate quickly splits into 2 3-carbon molecules, and this is called 3-PG, 3-phosphoglycerate. And that's carbon fixation. Okay, it takes the carbon dioxide from the atmosphere and it fixates it, it combines it with RUBP, and then it splits, and now it's a usable molecule in the Calvin cycle. Now, phase 2 is the CO2 reduction. What happens next? So in this phase, CO2 is converted to a carbohydrate. It adds electrons and energy to that CO2 molecule. Okay, so let's take a look at how this happens. So we can see number 3 there in the green. So that 3-PG molecule at the end of stage 2 in carbon fixation is reduced to G3P through a series of reactions which involve that ATP coming in and NADPH, as you can see as well, from the light reactions. And what happens once that 3PG is reduced, that leaves ADP and NADP plus, those lower energy molecules, to return back to the thylakoid. Because remember, these are required to then be reduced through the light reactions, and then they come back in to the Calvin cycle since the cycle is continuously going. So now we can see at the very bottom there, number 5, that one of the G3P molecules is set aside to be a building block for glucose, whereas the remaining 5 of the G3P continues on in this cycle. So I'm going to pause here for a moment because you're probably saying, wait, I thought only one G3P molecule is being made, right? We are following the path of one being made. However, I put stars beside these molecules that we're talking about, and those stars inside actually say the exact amount that's being produced. So if you look back at the top at stage 1, or not even stage 1, at the very, very beginning, look back up at the very top, 12 o'clock, at the CO2 coming in. Okay, right above the CO2, it's actually saying that there's 3 of them. So 3 CO2 is then creating, if you look at stage 1, it's creating 3 really unstable 6 carbon molecules. And you can see through the process, it's showing the actual numbers, so that 3 split, and now there's 6. Okay, and at the end, that's why we get, not the end, but at stage 5, we get the 6 G3P molecules from 3 CO2s coming in. Just to confuse a little more, the auditing of the products here is a little tricky, but that's why I highlighted those with the stars to remind ourselves that that's what's happening, okay? So from the 6 G3Ps that are being produced from the 3 carbon dioxides coming in, only one G3P, which is the building block of sugar, gets released. The remaining 5 continue on into the regeneration stage. So stage 6, or step 6, we see through the use of ATP coming in, the remaining 5 G3P molecules are transformed into 3 RUBP molecules. And this RUBP can then be used to start the cycle once again to generate more G3P building blocks. Phew! Okay, that is the cycle. Now let's talk about how many times this cycle has to turn to be able to produce one glucose molecule. At the top, I put an asterisk right beside the CO2. It's a nice blue asterisk there at 12 o'clock of the cycle. I wanted to remind ourselves that this cycle releases one G3P molecule for every 3 turns. There's 3 carbon dioxide that is required to produce one G3P molecule. So it takes 3 turns, each turn taking in a carbon from that carbon dioxide, right, through the fixation. That's why it takes 3 turns to make that one G3P molecule. If you see the G3P molecule, it has 3 carbons, right? So it needs 3 carbon dioxides, 3 turns to finally make that one G3P molecule. Then the cycle, so we have one G3P molecule, we need to double that amount to make 6 carbons for glucose. So to make one G3P, it takes 3 turns to fixate 3 carbon from the carbon dioxide. So to double that, it needs 6 turns to make that one 6 carbon glucose molecule. Take that in. You might have to revisit this page and mull it over to finally get those numbers to kind of click, okay? Now, to make one G3P molecule that is released, what do we need in terms of energy? This is an exergonic reaction. It needs to take in energy. How much do we need to make one G3P molecule? So if we look through this process, this whole process is showing the creation of one G3P molecule. Okay, it's kind of combining the 3 turns into one because it's showing 3 carbon dioxide coming in at once. So if we add up that 6 ATP just after stage 2, step 2, okay, we see 6 ATP coming in. This is the CO2 reduction phase. So we have the 6 ATP there. We also have 3 ATP when required for the regeneration of RUBP after step 6. Okay, so 6 plus 3, that gives us 9 ATP. We also need 6 NADPH. So you can see the 6 NADPH also coming into play when CO2 is reduced around stage 3 and 4. Okay, so this is the energy requirement to make one molecule of G3P that is released. But when we're talking about an entire glucose molecule, remember, we have to double that. We need 6 carbon, not just 3. So we'll multiply that 9 ATP by 2. We'll multiply the 6 NADPH by 2, right? Because we're talking about a whole glucose molecule. And the energy requirements for an entire glucose molecule to be created is, in fact, 18 ATP and 12 NADPH for that one glucose molecule.
And we're not strangers to G3P. We've actually seen G3P already. Maybe in your head you said, hey, that rings a bell. How come I remember the compound G3P? because this was produced in a step in glycolysis, right? As an intermediary step on the way to being reduced to be pyruvate. So similar to glycolysis, but it's backwards. We're not breaking it down. In this case, in the Calvin cycle, we are building it up. So to G3P and beyond, ribosylaldehyde 3-phosphate is the end product of the Calvin cycle. This is the building block that the Calvin cycle creates to create that glucose molecule. It's an energy-rich 3-carbon sugar. Okay, you can see that it was reduced, and through reduction, it gains more energy because it bonds. It creates more bonds. Okay, and because it's a 3-carbon sugar, sometimes you may see this being called 3C photosynthesis because it's actually synthesizing the 3-carbon sugar that eventually bonds together to form larger compounds. G3P is a very important intermediate. We have a whole page about G3P right now, so that tells you the importance of it. So we talk about G3P coming together to form glucose molecules because glucose is the main compound that often gets produced through photosynthesis. And glucose can make many different complex carbohydrates. But because it's a synthesis of carbon compounds, that G3P molecule, it can also contribute to the formation of lipids. And we know that lipids is a category of phospholipids. It can create fats, it can create waxes, amino acids as well, which can create proteins, nucleic acids that can create DNA and RNA. So G3P is a very, very important compound. Not only does it make that really important compound of glucose, but it can generate many other different macromolecules required for that plant to sustain all of its living functions. Now remember Rubisco. Rubisco you saw in the top, that first stage there. And this is the enzyme which fixes carbon from the air. It's really important. Rubisco is short for ribulose biphosphate carboxylase, which is why we like to just call it Rubisco. And many would say it's the most important enzyme in the world. Okay, there's actually quite a few important enzymes without other ones, such as the one that unzipped DNA. We wouldn't be getting replication and reproduction of DNA, right? But when we're talking about the photosynthesis, this is at least the most important enzyme in photosynthesis. It makes life out of air. It's taking that carbon dioxide and enables the cell in the stroma of the chloroplast to synthesize carbohydrates. And it's the most abundant enzyme as well. So let's look back at the accounting. I know I had that on the page that told us everything at once, but let's just recap. 3 turns of the Calvin cycle brings in 3 carbons from the carbon dioxide. So that generates 1 G3P molecule, right? So 3 carbon dioxide makes 1 G3P molecule, which has that 3 carbons in it. So then 6 turns of the Calvin cycle, right, double that, creates C6H12O6 when we're talking about the formation of glucose. So we need 6 turns total. So that means when we're talking about carbon dioxide, it takes 6 carbon dioxide, right? It has that 6 carbon in there to make the 6-carbon sugar, C6H12O6. In the end, it's 18 ATP, right? 9 ATP for each 3 turns of the for the carbon to come in. 12 NADPH, right? Because it takes 6 NADPH per 3 turns. So 18 ATP, 12 NADPH to create that 1 glucose molecule. If there happens to be any ATP left over, remember, this is theoretical yield and use of energy, but doesn't actually mean in reality sometimes. So if there is ATP left over from the White reactions, it will be used elsewhere by the cell. Here is an overview of the White reactions. It produces ATP, it produces NADPH, and it releases oxygen gas as a waste product. We went over it many times, but this is a great little summary. The Calvin cycle, it builds that sugar. which is most often glucose, it uses the ATP and NADPH in the light reactions. It recycles ADP and NADP back into the thylakoid membrane for the light reactions to occur. Let's put this all together. So plants make both energy, ATP and NADPH, and sugars. And you can see putting it all together gives us that beautiful equation of CO2 plus H2O plus light energy reacts to create that glucose molecule and that output of oxygen gas.